Ever found yourself pondering about how substances move inside a cell? Well, hold on to your microscopes, folks, because we're about to embark on a journey into the intricate world of cellular transport. Imagine, if you will, the bustling city inside every cell, where goods have to be moved, processed, and exchanged every second. Sounds familiar? That's because it's not so different from our everyday activities like eating, drinking, and even breathing. In this bustling cellular metropolis, there are different modes of transport. Some substances take the easy route, cruising into the cell without a care. Others need a bit of a push to get them moving. Then there are the heavy lifters, moving substances against the tide. And let's not forget the in and outers, who are all about exchange. So, are you ready for an adventure? Dive with us into the microscopic world of cells and uncover their transport secrets. Imagine hitching a free ride. That's what substances do in passive transport. It's like the lazy cousin of cellular transport methods, where substances don't really need to put in any effort. They just go with the flow. Let's dive into it. First up, we have simple diffusion. Picture this. You just dropped a sugar cube into a piping hot cup of tea. No stirring necessary. The sugar cube starts to dissolve, and eventually the sweetness spreads throughout your tea. That's simple diffusion in action, folks. Substances moving from an area of high concentration, like our sugar cube, to an area of low concentration, the rest of the tea, until there's an even spread. Now let's talk about its more complex sibling, facilitated diffusion. Imagine you're at a concert and the crowd is thick. You're trying to get to the front, but it's a struggle. Suddenly, a kind-hearted security guard takes you by the hand and guides you through. This is what facilitated diffusion is like. Some substances can't cross the cell membrane on their own. They need a little help from carrier proteins, our friendly security guards. Again, it's all about moving from high to low concentration, but this time with a little assistance. Lastly, let's talk about osmosis. Think of a thirsty sponge soaking up water. Water molecules move from where they're abundant, outside the sponge, to where they're less abundant, inside the dry sponge. In the cell, water moves through a special type of protein channel called aquaporins, from an area of low solute concentration to high solute concentration. It's nature's way of balancing things out. So, in passive transport, substances just go with the flow, like chilled beachgoers. They don't need energy or a ticket to ride. They're the easy riders of the cellular world, making their way through life one diffusion or osmosis process at a time. Stay tuned to learn about the go-getters of cellular transport in our next scene. Ever felt like going against the crowd? That's what substances do in active transport. Now, imagine you're a determined salmon swimming upstream against the raging current. The water's pushing you back, but you're a stubborn one, aren't you? You muscle your way up, battling the odds, defying gravity. That's exactly how active transport works, folks. It's like the determined salmon of the cellular world, always going against the current, or in this case, the concentration gradient. Active transport is the process that moves substances from an area of low concentration to an area of high concentration. Now, I know what you're thinking, but that's the opposite of what things naturally want to do. And you're right, it's like inviting a cat to take a bath. It just doesn't happen without a little persuasion. But why, you may ask? Would a substance want to go against its natural inclination? Well, let's say you're at a party and everyone's crowding around the snack table. You want to get some of those tasty treats too, right? But there's no space. So you decide to move to the less crowded area, where you can chow down in peace. That's essentially what substances are doing in active transport. They're moving from the crowded area to the less crowded area, even though it takes extra energy. And where does this energy come from? It's all thanks to a molecule called ATP, or adenosine triphosphate. This little guy is like the fuel for our determined salmon, giving it the energy to swim against the current. ATP provides the energy required to move substances against the concentration gradient, making it possible for the cell to get the materials it needs. So, next time you're battling your way through a crowded room or swimming against the tide, remember the determined salmon. Remember active transport, the process that defies the norm, that goes against the grain, using energy to move from low to high concentration areas. It's a tough job, but someone's got to do it. Active transport, the tough guy of the cellular world, always going against the grain. Ever tried moving a sofa all by yourself? That's what cells deal with in bulk flow and dialysis. Now, if you've ever seen movers expertly pack up a house and transport everything to a new location, you have an idea of how bulk flow and dialysis work. They're like the heavy lifters of the cellular world. handling the transportation of larger molecules and ions. So, let's get to know these heavy lifters a little better, shall we? First up, we have bulk flow. This is a process that moves large molecules like proteins around the cell. It's like a moving truck, capable of carrying hefty loads from one place to another. But unlike a moving truck that needs a driver, bulk flow is driven by pressure differences. When there's more pressure in one area of the cell than another, molecules get pushed from the high pressure area to the low pressure area. Next up, we have dialysis. Now, you might have heard this term in relation to kidney treatment, but it's also a cellular process. Dialysis in cells is all about balance. It helps maintain the right concentration of ions and molecules. Think of it like a meticulous packer, making sure everything is just right. It separates smaller molecules from larger ones using a selectively permeable membrane. The small molecules pass through while the larger ones stay put, like sorting out your socks from your sweaters in packing boxes. But what's really cool about these processes is that they're always on the go, working tirelessly to keep our cells functioning smoothly. They're the unsung heroes of cellular transport, always ready to step in when the cell needs a heavy lifting job done. So the next time you're struggling to move a heavy piece of furniture, spare a thought for your cells. They're doing this kind of work all the time, and they don't even break a sweat. Bulk flow and dialysis, the heavy lifters of the cell, always ready for some serious lifting. Ever had to squeeze through a tight spot? That's endocytosis and exocytosis for you. Now imagine a bustling nightclub with a steady stream of people going in and out. Our cell is just like that club, and endocytosis and exocytosis are its diligent bouncers. Endocytosis is like the bouncer who lets people into the club. It's the process by which cells absorb molecules, such as proteins, from outside by engulfing them. Picture this: a VIP, let's call him Protein, shows up at the club. The bouncer, endocytosis, recognizes him and lets him enter by wrapping him in a part of the club's wall, which then forms a separate room, or vesicle, inside the club. On the other hand, exocytosis is the bouncer who ensures that people leave the club when it's time. It's the process where cells expel waste and release molecules such as hormones and neurotransmitters. It's like when the club is closing and the bouncer, exocytosis, escorts the unruly patrons, let's call them waste, out of the club. The bouncer guides them to the door, the cell membrane, and then gently but firmly pushes them out into the night. These two processes, endocytosis and exocytosis, are vital for maintaining the integrity of the cell. They help in keeping the cell clean by removing waste, and also ensure that essential substances like nutrients and proteins make their way into the cell. Without these processes, our cells would be like a club with no bouncers, chaotic, disorganized, and downright unhealthy. So next time you think of a cell, don't just imagine a tiny blob. Instead, picture a bustling club with its own bouncers, endocytosis and exocytosis, working tirelessly to keep things in order. They might not be the most glamorous part of the cell, but they sure are crucial. And that, my friends, is the beauty of the cellular world. It's a place where even the smallest processes have significant roles and everything happens for a reason. Endocytosis and exocytosis, the cell's very own bouncers, always keeping an eye on the door. From hitching free rides to lifting heavy loads, cells sure know how to get things moving. Our cellular world is a bustling metropolis, with molecules and ions zipping around in a well-orchestrated dance. The easy riders, also known as passive transport mechanisms like simple and facilitated diffusion and osmosis, scoot around without spending any energy. They're the cool kids, always going with the flow. Then we have the go-getters, the active transporters, who aren't afraid to spend some energy to move against the concentration gradient. They're the ambitious ones, always striving for equilibrium. Let's not forget the heavy lifters, the masters of bulk flow and dialysis, moving fluids containing solutes en masse. And finally, the in and outers, endocytosis and exocytosis, the cellular equivalent of eating and throwing up, respectively. They're the maestros of large molecule transport. And that, folks, is how the microscopic world of cells manages its transport, a symphony of movements, each playing a unique yet harmonious role.